All right, thank you for coming to today's seminar. It's a pleasure to have Toshio Tsukiyama here today from the Hutch. I sort of looked through Toshio's CV to find some interesting or relevant tidbits, and I discovered that Toshio was actually trained as a vet in Japan before he kind of was corrupted and became a scientist. But he eventually did, and he after getting his DVM, he got his PhD from Hiroshima University studying sort of molecular biology of various viruses. And after that, he kind of pursued his interest in molecular biology by doing a postdoctoral fellowship in Carl Wu's lab at the NCI, which is perhaps appropriate since today's seminar is sponsored in part by the PSOC, which is sponsored by the NCI. While he was in Carl's lab, he did some really beautiful biochemistry, purifying and characterizing the mechanism by which ATP-dependent chromatin remodeling complexes work. And in the course of that work, discovered the sort of founding member of a chromatin remodeling complex family called the imitation switch or ISY family, which sort of became the focus of his own research when he joined the Hutch as a member in 1997. Since then, he's done a lot of really nice work on the ISY complex and really trying to understand both mechanistically and in terms of the biology of ISY and how it works at a genome-wide level, which will sort of be the focus of his talk today. While he's been an independent investigator, he's been awarded both a Pew Scholarship and a Leukemia and Lymphoma Society Scholar Award. And he's done extremely well. And so with that, I'll pass it off to Toshi. The title of his seminar is Control of Non-Coding RNA Transcription, and DNA Replication through Chromatin Regulation.
Thank you, Jason. Can everybody hear me? Great. Yeah, I can open animal clinic technically tomorrow in Japan, but I have no clinical experience, so don't ask me pet questions. Unless you don't care about your pet. Yeah, so I used to work on animals, then mammalian tissue culture as a grad student, then Drosophila as a postdoc, now yeast. So I'm going down evolution trees. So next is bacteria and all in vitro before I retire. Anyway, I'd like to share with you, thank you very much for coming, and thank you, Jason, for inviting me. I've been having a great time here. I've never been to Northwestern before, so it's been a great experience. And I'd like to share with you today a couple of ongoing stories going on in the lab regarding chromatin biology. And hopefully, everybody in this room knows that in eukaryotes, genomic DNA is compactly stored in the form of chromatin, which allows huge condensation for compact storage. However, at the same time, this condensation causes huge problem for cells when they need access of transacting factors to specific sites on DNA for DNA-dependent processes. And even the most fundamental unit of chromatin, the nucleosome, single nucleosome, which I'm pretty sure I don't have to introduce, has ability to impose huge restriction for accessibility. Now, because of this, we now think that chromatin structure, understanding chromatin structure is essential, because essentially in understanding anything that takes place on DNA, because it affects accessibility so profoundly. So the long-term interests in the lab are twofold. One is to understand really how chromatin structure affects DNA-dependent processes in vivo. And in turn, we also would like to understand then how then chromatin structure is regulated to accommodate these DNA-dependent processes. And currently, in recent couple of years, our focus have been on two DNA-dependent processes, especially on non-coding RNA transcription and DNA replication, because we think that those two DNA-dependent processes have not been studied enough in terms of connection to chromatin structure, and there's a lot of interesting biology to be learned. So today's my talk concerns both of those projects in the lab. As you'll see, both are reasonably still at the beginning and pretty early in the stage, but we think there's something interesting happening in both areas. So the first part of my talk is about
chromatin structure and cryptic RNA transcription. And all the data I'll show you, all is unpublished, is produced by a talented graduate student, Adam Yeden. But this project was initiated by two former members of the lab, Dan Vandermark and Easton Whitehouse. Now, this line of work, as Jason introduced, was basically continuation of our long-term interest in understanding the in vivo functions of I-switch or imitation switch ATP-dependent chromatin remodeling complexes. As shown as the schematics here, this class of chromatin remodeling factors, all of which are ATPases, form multiple subunit complexes, and the complex is evolutionarily highly conserved, which suggests a possibility that they play some interesting, important roles in vivo. And consistent with that, we have learned over years that I-switch remodeling factors are essential for development in Drosophila, C. elegans, and mouse. And in Drosophila, it's essential for cell viability at the single cell level. And probably the coolest phenotype of I-switch loss is in Drosophila. If you reduce activity of I-switch in cells, you can see that the entire chromosomes become puffy for all the chromosomes, showing that I-switch is somehow required for integrity of chromosome structure at the mass level. In turn, in vitro biochemical analysis of I-switch complexes have been pretty well advanced. And every single factor we know that's been tested has ability to slide nucleosome along with DNA without changing histone DNA contact that much. And some of those factors can space nucleosomes from irregularly spaced one into regularly spaced one, or facilitate assembly of nucleosome arrays, or some of them can exchange histones within established nucleosomes with the ones that's freely available. Now, current challenge in the field has been really to connect the dot between this and this. That is to really understand how those biochemical activities are utilized in vivo to affect specific DNA-dependent processes. And that's what we have been trying to do in the lab. And today's talk is regarding one of the two I-switch ATPases in the Saccharomyces cerevisiae, I-switch 2, which, as you can see, has highly conserved subunit structure throughout evolution. Now, the next slide summarizes basically what we learned in the first several years of work in the lab. We learned that I-switch 2 is targeted to several classes of loci. One is promoter regions of subset of genes. The other is upstream regions of tRNA genes. When I-switch 2 is targeted to those regions in accessible or open chromatin structure, somehow it slides nucleosomes in one direction to close the gap by converting open or accessible chromatin structure into closed or inaccessible chromatin structure. As a result of this I-switch 2 dependent chromatin remodeling, we know that transcription of certain class of genes are repressed, and upstream of tRNA regions, integration site selection of TY1 retrotransposon changes as a result of this change. Now, a few years ago, we had a big surprise that we were missing one very big class of I-switch 2 targets. We found this when we were mapping nucleosomes genome-wide in wild-type and I-switch 2 mutant. This summarizes our finding. It turned out that I-switch 2 is targeted to about 8 to 10% of the genes at 3' ends, which we didn't even know that exist. And when I-switch 2 is targeted to 3' ends of the genes, it slides nucleosomes in the downstream region from genic to intergenic region. Somehow, when this happens at 3 loci we tested, this prevents cryptic transcription from going into the wrong orientation. So as a result, if you lose I-switch 2, those nucleosomes slide off this way, and this somehow permits non-coding transcription to initiate in this upstream orientation, which is very interesting. So when we submitted this paper, all the referees asked us to look into this more closely, how frequently this happens, what the biological meaning of this might be. And of course, we were very interested in that, partly because, as you know, it's become clear in recent years that eukaryotic genome is transcribed all over the place, from yeast to humans. We know very little about the biology of those, how they're regulated, and what the biological functions of those non-coding transcripts might be. And we thought we might be able to contribute to this. So after this paper came out, one of the focus in the lab has been to really understand more about the biology of ICH2-dependent effect on non-coding transcripts. The first thing we decided to do then is to look at the correlation between initiation of those transcripts with chromatin structure or nucleosome positions. Here, I'm showing one ICH2 target, where ICH2 is enriched at 3' end of this gene and slides nucleosomes. And those are two nucleosome mapping data on high-density arrays versus gel-based traditional southern blot-based nucleosome mapping. As you can see, the correlation is extremely good. We can get almost one-to-one correlation between those peaks. Of course, that's why I'm showing you this data. But because of that, we can map nucleosome positions pretty precisely in wild type and ICH2, showing that this complex, when it's targeted here, slides nucleosome this position into this position. Now, when we did prime extension to identify the start site for non-coding transcripts in ICH2 mutant, we were able to map initiation sites around here, which is satisfying because this is exactly where this complex changes nucleosomes. The thing that caught our eyes, though, is those two bands. This is because, as you can see, the distance between those two bands is just too short to accommodate regular nucleosome. On array data, you can see that protection against the nucleus we use for mapping nucleosome is a lot less robust compared to canonical nucleosome. Because of this, we started to wonder if this corresponds to NFR or nucleosome-free regions. And NFR, the concept of nucleosome-free regions at promoters is actually pretty old. But as far as we know, the genome-wide mapping of NFR was done in 2005 by Oliver Randall's lab. Here's one of the data from their paper. What they did was to map nucleosomes using high-density arrays along with chromosome 3, which has over 200 genes, and see where nucleosomes are. And what they did, in this case, both red and green are nucleosomes. In silver are linker regions. And what they did was to align those nucleosomes at the promoter of over 200 genes such that this is the first nucleosome in the transcription unit. We call this +1 nucleosome. And each horizontal line corresponds to each locus. So this is about 200 genes. When you align nucleosomes in this way, I think it's very clear that the linker region between this nucleosome and this nucleosome is wider than other places. This is how they originally identified NFR. Later, a couple of years later, Frank Pugh's lab has mapped nucleosomes around the promoters by mapping sequencing nucleosomes containing histone variant HTZ1, and then also found that this is a true genome-wide. When we did the nucleosome mapping in the same year, 2007, we saw the same thing. In this case, the red is nucleosome, the green is a linker region. What we did was to align nucleosomes around the promoter of basically all yeast genes that we were able to map. And then what we are doing here is to align those nucleosomes in the middle of the first nucleosome in the transcription unit, which we call is +1 nucleosome. When we do this, it's very clear that the distance between the +1 nucleosome and the next nucleosome upstream, -1 nucleosome, varies quite a bit. But in general, this distance is a lot wider than other places. And genome-wide average is shown in here, and there are about 6,000 lines here. So this also annotated NFRs in this way. What became clear in the last few years is that NFR is a really interesting hot regions in terms of transcription regulation for mRNA genes. For example, in yeast, over 4,000 genes transcription start site have been mapped. If you see where things go in this alignment, as you can see, it's here, meaning that the vast majority of transcription initiation, at least for mRNA genes, start at the left side of the +1 nucleosome. It turned out that this having nucleosome depleted the area around transcription start site is ubiquitous among eukaryotes. This is true from yeast to Drosophila to humans, even though actual distance between NFR edge and start site might vary depending on the species. And also, if you look at where transcription factors bind in this alignment, you see something pretty stunning. On the right, this is a transcription factor binding sites for virtually all DNA binding proteins on yeast genome, and this is overlay, and white signal means binding site. As you can see, very significant fraction of transcription factor binding occurs over or around the NFRs. And also, I'm not going to show you the data, but histone variant HTZ1 has been shown to paint NFRs either side. So all of these results strongly suggest that this is basically the center of the universe in terms of transcription regulation in yeast and in other eukaryotes as well. And how then this NFR structure is maintained or regulated becomes very important in understanding transcription. Now, coming back to this data, the reason why we got excited about looking at this data at the end of 2007 was because this then suggested the possibility that not just for mRNA transcription, but even also for non-coding or cryptic transcripts, maybe NFR tends to be the initiation site. Maybe this is a universal start site for transcription for both non-coding and coding transcripts. And then furthermore, this suggested the possibility that one of the major functions of I-switch is to be targeted around NFRs to regulate this kind of a transcription. So this became our model, that is whether I-switch2 is targeted around NFR specifically to suppress non-coding RNA transcripts. That's what Adam decided to tackle on. Of course, the first thing we needed to do was to annotate NFRs genome-wide, which was not done when we started this project. And originally, we thought this is a trivial project because there are several high-quality nucleosome mapping data available in the field. However, it turned out to be actually quite a bit of pain. Main reason is because even though overall agreement of nucleosome mapping data, 2 by deep sequencing, 2 by high-density arrays, agreement is pretty good. Every single data set has regions where nucleosomes could not be mapped very precisely for whatever reason. There's just no way that nucleosomes are completely missing from these wide areas. But every single data, whichever method you use, there is a very significant fraction of genome that behave like this. So because of this, if we use simply the distance between nucleosomes to map NFRs, those regions dominate our data set. And we needed to come up with an algorithm to throw away those problematic regions. So I'm not going to go into the detail, and I'm happy to discuss how we did this later if you're interested in. But we basically demanded that around NFRs, nucleosome signal have to be robust. When we do that, we are very conservative, and I'm pretty sure we are underestimating the number and width of NFRs, but it was surprising for us to find that there's a lot of NFRs throughout yeast genome. Interestingly, this number is very close to the number of total genes on yeast genome. And this, even though this is very conservative estimate, this occupies about 5% of the yeast genome, which is very surprising to me as well. And because of this, because though we are able to identify them in unbiased fashion, depending on the proximity to protein coding genes, we were able to break them down into 4 classes. The ones that are close to 5' ends with the genes, 3' ends. And interestingly, surprisingly, actually, there's a significant number of NFRs that are buried completely within open reading frames. We actually don't know the function of them. And also significant fractions are within long linker regions, long intergenic regions that are not close to protein coding genes. And because yeast genes have very compact genome, a lot of genes actually share those NFRs, and breakdown is shown in here. So the question then, after annotating NFRs to test our hypothesis, we wanted to see whether I-switch2 targets are in close proximity to NFRs, and the answer is yes. The number is a little old. We need to update this, but it doesn't matter. This is a really significant overlap. So very significant fraction of I-switch2 targets are within the nucleosome distance from NFR. This established the first part of our model that major function of I-switch2 is to be targeted around NFR. So what happens to the chromatin structure? So we broke down I-switch2 targets into 4 different classes. So it's a little bit busy, but each column is NFR in the middle and plus minus 300 base pairs. And yellow signal is positive. Sorry, we switched the color for colorblind people because this is easier to see. And it doesn't matter which class you see. As you can see, ISH2 is very preferentially targeted around NFR. Now, interestingly, a lot of cases, it's targeted to one side of NFR. When that happens, and when we look at the places that nucleosome position changes in yellow again, we see corresponding changes in nucleosome position. So it doesn't matter which classes, even within open reading frame, ISH2 is targeted to NFR. When we see what happens to nucleosome positions, on average, this is what you see. It might be a little hard to see from the distance, but the basic bottom line is in wild type, the distance between those nucleosomes is narrower than in ISH2 mutant, showing that for all the classes, the function of ISH2 is to reduce the size of NFR. And as it shows you, there are many transcription factors that bind NFRs, and all of those factors that tested so far are shown to work positively for NFR size. All of them increase accessibility of NFR, and we believe this is only the factor known so far to negatively regulate the size of nucleosome-free region. So how about the cryptic transcripts, which is another side of our model? So it turned out that analysis of cryptic transcripts is really painful, partly because there's an exosome that's highly conserved from yeast to human, which is a very abundant, very active exonuclease, whose main job is to do RNA surveillance to rapidly detect cryptic transcripts and polyadenylate and degrade them. And because of this, when you have full activity of exosome, it's very hard to detect cryptic transcripts. I show you one example. And this is where ISH2 is targeted. When we did strand-specific northern blot, in the presence of full activity of the exosome, we see basically no antisense transcripts. However, if we just reduce a little bit the exosome activity by deleting a non-essential component of the exosome pathway, TRF4, suddenly we see huge stabilization of antisense transcripts. Then addition of ISH2 mutations significantly increases this. We did this kind of analysis at several loci, and majority of places behave in this way, meaning that unless you reduce exosome activity, there's no way you can analyze cryptic transcripts. You basically totally miss out. So because of this, we decided to use two mutants that are not essential but been shown to stabilize cryptic transcripts, TRF4 and RRB6. And also because we have no idea how those cryptic transcripts are processed in vivo, especially in the context of those mutants, we wanted to be able to detect those transcripts without any selection or amplification. So we needed highly sensitive detection system. That's why it took us a little longer than we wanted. But we use custom tiled arrays right now for detection, and we tiled both strands of 3 chromosomes that are biologically interesting to us, chromosomes 3, 6, and 12, which covers about 14% of the genome. Now, a definition of cryptic transcripts are those that are more stabilized in those mutant background. And of course, in addition to that, we delete ISH2 to see what happens. So this is one of the examples of how our data looks like. Ooh, do I need to dim this? Can you see it? Okay. Oh, great. Thank you. So this yellow means RNA signal. In this locus, there are a couple of genes going from left to right, and all of those genes are going from right to left. And we're looking at the top strand. As you can see, just by deleting TRF4, we see huge stabilization of RNA in this region, which is antisense to all of those genes. We see the same thing in here. By deleting ISH2 on the top of TRF4, we see this even going farther and here. And it's actually the same thing here and here as well. So this happens very frequently. And this is a breakdown. Thank you. And as you can see, this covers only 14% of the genome, but there are more than 500 loci where we can detect cryptic transcripts, meaning that genome-wide, there are thousands of loci that this is happening. So it was pretty surprising to us. When we see how those regions are initiation sites close to NFRs. We see, even though this is a very conservative estimate, we see significant fraction of them are actually initiating close by to the nucleosome-free regions, which is consistent with what's published last year by other groups. When we look at ICH2-dependent de-repression of cryptic transcripts, we see that number of loci we detect those is a little bit lower than TR4 single RRP6 mutant, but still we are looking at hundreds of loci genome-wide. And again, significant fraction of them initiate around NFRs. Just to reiterate how ICH2-dependent chromatin structure, regulation, and non-coding RNA transcription are correlated, we align all the genes in a way that this is the sort of the beginning of where we detect cryptic transcripts, and all the genes are oriented in a way that cryptic transcription goes this way. And see what happens to chromatin structure in wild type and ICH2 mutant. As you can see, you see very dramatic shift in nucleosomes. So this is a wild type, and this is a mutant, meaning that in wild type, in the presence of ICH2, this complex is sliding nucleosome from this way to this way towards the start side of a transcription to repress transcription. So these results establish that our model is correct, that one of the major functions of ICH2 is to be targeted around NFRs, and part of the reason is to do this, to repress non-coding transcripts. The next question was why you have to do this. Of course, it's easy to imagine that you just don't want to waste resources to transcribe RNA just to be degraded. And if they escape the degradation system, it can produce non-functional or even toxic proteins. But the question is whether there's something else. The first clue came from the fact that each genome is so compact that the majority of those cryptic transcripts we identify in those gene pair, mutant pair comparison is overlapping with mRNA genes, coding genes. Only those shown in dark blue are the non-coding transcripts that are not overlapping with mRNA coding genes. So because of this, we wondered whether sometimes increased cryptic transcription can cause transcription interference with mRNA, and we have evidence for that as well. Okay, so this is actually the same locus that I showed you, where TR4 mutation or ICH2 mutation increases non-coding transcripts. If you look at the other strand, which is sense transcripts for those guys, you see almost a mirror image. That is, when non-coding transcripts go up, coding transcripts go down here. It's actually true here as well. And whenever we see this, those non-coding transcripts almost always go through the promoter of those genes, suggesting a possibility that the way this happens is by non-coding transcripts knocking off transcription apparatus for the coding transcripts. And if you are curious to see how often this happens, in TR4 single mutant, our estimation is this happens at hundreds of loci genome-wide. And in ICH2 dependent way, this happens in the order of tens throughout the genome. So it's pretty frequent. We also see that interference happening in the same strand as well. Here in TR4 mutant, we see cryptic transcripts hugely stabilized, and then coding transcripts of this gene going down. This is a little less frequent compared to the antisense interference, but this, again, is happening probably in the order of tens. So it's pretty common as well. So based on these results, our feeling now is that one of the major functions of ICH2 is to be targeted to NFR. And this function is a unique factor to reduce the size of NFR. And the result of this is suppression of transcripts in both coding and non-coding way. And the reason why it has to do that for non-coding RNA is, at least in some cases, this reduces the transcription interference with coding transcripts. Now, we are doing a lot of work now trying to understand more about the biological meaning of these transcripts, and also more broadly, why non-coding transcription happens in this way. Because to me, it's actually the most interesting question because it appears almost like RNA polymerase is so non-specific that whenever there's a gap in nucleosome, it comes in and starts transcription this way or this way as it feels like. And of course, it's possible that it's very hard to design RNA polymerase to be so specific in during evolution. But it's a little hard to imagine that those non-coding transcripts don't have any biological functions that we are not aware of. So that's one of my personal favorite. I'm trying to persuade Adam to be excited about this project. If he doesn't, I'll start working on that myself. OK, so this is my first part of my talk. If you don't have any questions, I'm going to move on to the second part of my talk. OK, so the second part of my talk is totally different. This is about DNA replication and histone modification. And this work was done single-handedly by a very talented former graduate student in the lab, Ashwin Unicreation, who graduated end of last year. Now, this is something that I got personally interested in a while ago, several years ago, around the time Ashwin started grad school in my lab. That is how histone modifications can affect DNA replication. Now, if you're familiar with the chromatin field, you probably appreciate that there's really not much known about chromatin regulation and DNA replication, although chromatin structure should affect every DNA-dependent processes. So we wanted to tackle on this. And indeed, there are many possible ways DNA replication can be affected by histone modification, just like transcription. Just like transcription, it requires multiple protein binding to specific sites and huge RNA polymerase going through chromatin. And also, chromatin assembly can be linked to replication fork progression. If that's the case, histone modifications can affect replication in that way. And of course, it can affect replication indirectly. And of course, there are actually a lot of papers in the field strongly suggesting that histone modifications, especially acetylation, strongly affects replication. For example, if you delete major deacetylase RPD3, you can accelerate the timing of origin firing throughout S phase. Or if you delete another class of histone deacetylase SER2, again in yeast, you can increase the probability of any given origin to fire in any given S phase at ribosome DNA locus. However, there's really not much known beyond this. That is exactly which modifications on which histones matter for replication. And moreover, for people like us who are really interested in the mechanism in vivo, which steps are affected by those modifications. And even after that, how those modifications affect that specific step in DNA replication. Those things are almost entirely unknown. And of course, this was totally new project to me and to us. And we wanted to start this project in unbiased way, if possible. And of course, we were aware of the fact that there are many, many very good genetics lab trying to identify all the factors involved in DNA replication through large number of genetic screens. So if we, and I'm not the geneticist by any training, so if we start genetic screen, the chance of finding something important new is pretty low. So we wanted to do something totally different that's never been done before. And I'm a biochemist by training. I love purifying protein. So we decided to take biochemical approach and very simple-minded. That is to purify histones that are specifically present around active replication origin throughout the cell cycle, and just determine all the histone modifications that we can detect around origins. And then if we were able to find modifications that are present only around S phase or only absent in S phase, or if we find a modification that abundance changes specifically during S phase, maybe they are good candidates for those that are involved in replication. Sounds simple. OK, so we needed to first determine how we detect modifications. This was easy decision, because in 2003, Mark Potham's lab showed off us how useful mass spec is as a detection tool. And only several years ago, this was basically all the modifications known at the time using traditional methods, such as antibody-mediated method or by labeling. Then just by shooting half-thymus histone into high precision mass spec at the time, they were able to identify very large number of modifications that we didn't know existed. So we decided to use mass spec for detection. The problem is that at the time when we started this project, there's no way to isolate histones specifically from origins that with sufficient purity and yield for histone modifications. Remember, we are only interested in histones around replication origins. So we decided to start by setting up the system. So we immediately noticed that TRIP1 RS1 circle makes sense for us because this has been around for a long time, and it's only 1.6 kbs cell. And this RS1 origin behaves very well as a mini circle, and we know that this is regulated pretty much like endogenous origins. And also, Fritz Thomas' lab have mapped that there are only 7 nucleosomes on this template. Because of this, if there are histone modifications that are very rare and only present around active origins, we expect that those modifications to be enriched on histones on this template as compared to, say, genome-wide average. And the biochemical isolation of this circle has been pioneered by Bob Simpson's lab when they inserted a single copy of lock operator within RS1 and used immobilized loci as a module. What they did was to make whole cell extract from cells carrying this plasmid and run through the extract over this column after washing elute by IPTG. Now, this worked well enough for them to do Western analysis to detect proteins on the template. However, based on the experience of people who use the system, the yield and purity of chromatin is not even close to where we need it to be for comprehensive mass spec analysis. So Ashwin spent first couple of years of his graduate studies trying to maximize this method. I'm not going to go into the detail, but I'm happy to share with you the protocols because it was a lot of work and it works amazingly well. So we did a lot of optimization, and I love showing off this data. And this tells you how well it works. So by the way, name of the template is Talo8, trip RS8 copy, like, oh, oops. Then Talo8 purification, this is a gel. Okay, this is DNA gel, not a Southern blot. And this is starting material and unbound fraction, and this is Talo8. As you can see, we don't see genomic chromatin contaminating at all. Even when we did a Southern blot, we cannot detect genomic chromatin. So it's very, very pure. And importantly, each line corresponds to about 400 ml culture from low log phase, like OD.2. So yield is extremely good. I have to say this is after one-step purification by flag tag. Then if you look at the protein, it's even more impressive. Each line corresponds to about only 250 ml culture, and histones are already the major components in the sample, and we know that much of those are real chromatin binding proteins. So because of this dramatic improvement of the system, now we are able to do single-step purification and go straight to mass spec. So you can go from cell to mass spec within a day. That is, if you don't care about having a long day, but Ashwin didn't, so that was good for me. All right, so our strategy is pretty simple. We do this from asynchronous culture, cells arrested in G1 or cells released into S phase or G2M, and then immunopurify Talo8 and do mass spec. Importantly, we wanted to have a genomic control, so we purified the bulk chromatin from the genome at the same stage of the cell cycle. And then wanted to compare this to this. If there are modifications that we detect only on Talo, they are good candidates for those that are specific around replication origins. And Ashwin found that for him to be able to do mass spec analysis several times for high confidence, you need about 2 micrograms of histones. For that, he needs about 10 liters. And of course, doing cell cycle synchrony for 10 liters is a lot of work, but it's doable if you're Ashwin. Okay, and then he did the survey for 6 modifications in all possible combinations, and Ashwin manually verified every hit that we report. Okay, and because our purification worked so well, the coverage of histones is amazingly good. In red are the peptides we were able to cover, and are the highlighted where we cut peptides by RGC. And in black are the peptides we could not detect in our mass spec analysis. As you can see, vast majority of those black peptides are just too short to be analyzed, meaning that we were able to detect most of detectable peptides. And this is a list of modifications we found, and as you can see, we detected a lot. And surprisingly, large number of modifications were specifically detected either only on TALO8 or, interestingly, on bulk only. And a significant fraction of them were detected only in cell cycle specific manner. And we're looking into function of many of those modifications. But today's talk is actually on modifications that we find all over the place. But it turned out those are pretty interesting. Now, one of the real power of mass spec analysis is that you can detect all the modifications on the same peptide in all the possible combinations. And this is very convenient for histone N-terminal tails where multiple modifiable lysines are present. And H4, for example, has those 4 modifiable lysines in close proximity, and we detected every single possible modification in combination. Now, what became very interesting was when we started quantitative analysis of histone acetylation throughout the cell cycle, we used a method that's developed by Dan Gottring, who is our colleagues. What you do is to take a mixture of histones, some of which are in vivo acetylated, shown in blue, then take this mixture and do in vitro chemical acetylation using deuterated acetic anhydride, which acetylates only the lysine that's not been previously acetylated, but they're chemically identical. So you go through mass spec, and then you can identify lysines that are in vivo or in vitro acetylated. And because they are different by 3 kilodalton, which is huge in mass spec world, you can basically compare the signals of those 2 peaks to identify the fraction of lysines that are in vivo acetylated. So we did that analysis, and this is the result. And I'm going to take you through this a little bit one by one. But we did the analysis in a few different ways. One is only on TALO8. This is bulk chromatin. And we can look at each lysine independently of other modifications on the same lysine, on the same peptide, or we can count number of modifications on the same peptide. Okay. And in red means more signals here. So let me first start with K56 of H3, which is known to be associated with newly deposited histones. If you look at that on both TALO8 and bulk chromatin, the level is very low in G1 and increases sharply in S phase. Importantly, to make my point, we see the same changes on TALO8 and bulk, which is consistent with the fact that this is a modification that comes with new deposition, because this implies that it doesn't matter how distant you are from origins. As long as folks go through your place and deposit new histones, you get these modifications in the same way. Okay, so this is consistent with the biology of K56 that we are aware. When we started looking into H4 tail, it was totally different. On bulk histone, surprisingly, there's little change in histone acetylation levels throughout the cell cycle, and it's dominated by K16. When we look at number of acetyl groups throughout the cell cycle, it's surprisingly, majority of acetylation is monoacetylation on the tail. This is a number. This stays around 40%, and about half of modification is K16. When we look at the TALO8, remember this is close to active origins, we see something totally different. As you can see, when you look at each individual lysine, level is very low in G1 and goes up very sharply in S phase. Then it's even higher in asynchronous culture. Because our asynchronous culture is 60%, about 60% G2M, and because level is low in G1, we can attribute majority of this to G2M. So it seems like for all the lysines we can detect, saturation level goes down sharply in S and even farther in late S or G2M. The real surprise came when we looked at number of acetyl groups. As you can see, monoacetylation level stays about the same despite this sharp increase in acetylation. It is the di, tri, and tetraacetylation that goes up very sharply throughout the cell cycle progression. Here's the number. As you can see, for example, tetraacetylation starts by less than 0.1% in our sample and goes up really a lot. So this implies that dynamic acetylation regulation happens on tau-8, which is nearby RS1. But this happens at the level of multiple acetylation per tail, rather than having more histones acetylated. In reverse, if you look at the difference between G2M, as inferred by asynchronous culture, and G1, again, we see very sharp decrease in acetylation. But this deacetylation event happens in a way that does not affect monoacetylation level. So for both acetylation and deacetylation, this happens strictly at the level of multiple acetylation per template. We are very glad to see this because otherwise, unless we use mass spec for analysis, it's very hard to detect this kind of regulation. And we believe this is the first case of dynamic acetylation regulation in operates at multiple acetylation per tail. And we see basically the same thing on H3 tails, even though it's not as dramatic as H4. So what the implication of this finding? I think when we say, oh, acetylation level changes or increases around Rs, origins around S phase, it can be either you're having more histones acetylated, or you can have small number of multiply acetylated tails. And our data clearly prefers this as an interpretation. And this is very important in thinking about the mechanism because it says that having multiple, even if it's small number, multiple acetylated tails around the origin sends completely different signal to cells as opposed to having many more histones acetylated at low number per tail. So how about actual functions in replication? Of course, tau-8 is very nice and neat, but this is artificial, and of course we knew that the referees will be unhappy if we stop there. So we needed to really test whether the multiple acetylation per tail we found is required for replication of endogenous origins. So as I said, H3 and H4 have multiple acetylatable lysines, and we saw all of this throughout the cell cycle. The question is it really the multiple acetylation per tail that's required? To address that, we did a systematic mutagenesis of acetylatable lysines. And to make a long story short, more acetylatable lysines you mutate, bigger the problem you are. So if you lose only 4 acetylatable lysines on 3 and H4, you're pretty much OK. You become a little bit sensitive to replication inhibitor. If you lose 5 lysines, you are in big trouble. And it's in bigger trouble if you lose 6. If you do cell cycle analysis of the mutant that lost 4 acetylatable lysines, you arrest the cells in G1, release into S phase to look at the S phase progression. You don't see much difference. Only if you have a little bit of replication inhibitor, you see detectable delay in S phase progression. However, if you lose 5 acetylatable lysine, only just 1 more, then you see in the absence of replication inhibitor, you see strong delay in S phase progression, which strongly supports our conclusion that multiple acetylation on H3 and H4 is essential for chromosomal replication. And this is not only HU, which depletes DNATP levels, but also another replication inhibitor, MMS. And the mutant that block histone acetylation are not sensitive to DNA damaging agent, showing that this is really specific phenotype. So we decided to use this quintuple mutant for further analysis. So the question then is, the next question that we are really interested in is which step in DNA replication, H3 and H4 acetylation, might be required. Remember, because we detected this multiple acetylation only on tau-8, which is close to active origin, we suspected that might be origin function. The first thing we did was to do plasmid loss assay using plasmids carrying either RE active origin, RS1, or late firing origin, RS14, 13. In both cases, if you compare wild type and histone acetylation mutant, the mutant showed significantly increased loss rate. And of course, this loss rate could be due to origin function, but it could be also due to something else like fork progression or plasmid segregation. So we next decided to employ a pretty cool assay that's developed by Doug Cochran originally. In this assay, you use 2 plasmids. One has, in addition to RS1, multiple functional origins. So the idea is if the elevated plasmid loss rate is due to failure in origin function, having multiple origins should significantly rescue that plasmid loss rate. Whereas if you have the same size plasmid that have mutated origins, you should still see elevated loss rate, because this gives you 8 times more chance than this plasmid to fire at least one of the origins throughout the S phase. When we transform those two plasmids into wild-type cells, loss rate doesn't change at all because one active origin is just enough. So that's good. If you do the same assay in histone acetylation mutant, however, having only single origin significantly increases the loss rate. And importantly, much of this increase can be reduced by having multiple origins. This very strongly supports the idea that it is the origin function that fails in histone acetylation mutant. So, and Ashwin didn't stop there and decided to look at the origin function directory by 2D gels. Because of interest of time, I'm not going to go into the detail, but he showed in the absence and presence of origin replication inhibitors, he was able to determine that origin firing efficiency goes down very significantly in histone acetylation mutant. Okay, so to summarize the second part of my talk, we were able to identify this extremely efficient method to purify a mini-circle in native fashion, which led us to discover that the histone H3 and H4 tails are regulated in a dynamic fashion throughout the cell cycle at the level of multiple acetylation per tail, which we believe is required for efficient firing of origins. And another thing that I wanted to make point at the end is that because this template works so well, if you're interested in other processes that can take place on this template, you are able to use this in multiple different ways. You can identify all the factors that interact with the template by doing mass spec, and at the same time, you can detect the modification status of all those proteins pretty easily. And because this works so well, you will be able to look at the template by EM to see what happens to the chromatin structure if you want. Or if you're interested in transcription, you can purify TAL08 and elute natively by flag peptide competition and use this for in vitro assays such as in vitro transcription or recombination. And you can use histone modifying enzyme mutants or histone mutants to let cells assemble chromatin in vivo, then use those templates in vitro. So I think the potential usage of this system is enormous, and we are more than happy to share this system and the protocol with anyone who's interested in this. And at the end, I'd like to thank people who contributed to the work. As I said, Ashwin did the second half of the work, and Adam did the first half of my work, my talk. And this is Mount Rainier, and this is a lake. See, we have sunny days in Seattle. And this is the hutch, and my lab is on the other side of this building. So we have a fantastic view of a concrete wall, but it's still a good place to do science. So thank you for your attention. Mm-hmm.
From non-coding RNAs, and they can interfere with the transcription from the other strand.
Right.
So are these things somewhat equal? If you can look at in the sensitized background, the normal level of non-coding transcription, you take Tata on Crypt, and the non-coding RNA is coming off of Watson. Does that increase the level of non-coding?
Oh, I see. Right? Well, the coding transcripts might be interfering with non-coding transcripts. That's an interesting idea. Yes. Oh, the question is whether I only talked about non-coding transcripts interfering with the coding transcripts, but whether the other way around might happen. That's a great question. We've never done that. So to reduce coding transcripts to see what happens to cryptic transcripts, yeah, it should be testable, and we should look into that.
The first part of the talk, what I switched to, I missed the mechanism targeting that.
Right.
Right. So the question is how ICH2 might be targeted to those 3' ends especially. That's a great question. Actually, until we got this result, we thought we knew how ICH2 is targeted because there are only several loci we knew ICH2 is targeted, is 5' ends. And those loci, handful of loci we looked at, can be explained by transcription factors. Now, there are several hundred loci, 3' ends that ICH2 is targeted, or some of those are within operating frames. So now we realize that we actually know nothing about ICH2 targeting. So that's another project that Adam is working on. So obviously, at 3' ends of the genes or within operating frames, we cannot explain targeting by transcription factor binding. But at the promoters, we have published several years ago that having nucleosome-free regions is one of the requirements for ICH2 to be targeted. So we are pretty certain that having wide linker region is part of the recruitment mechanism. But there are plenty of NFRs that ICH2 is not targeted. So we actually don't know. Yeah, we're wondering about that.
I have a question. So in the case of genes where there is a nucleosome-free region, upstream of the transcriptional start site, if you get rid of ISY, or actually I'll say it the other way around. If you activate transcription of those genes,
Right.
does non-coding transcription in the opposite direction, sort of upstream of the promoter, also increase?
Yeah, that's a good question. So whether, I think your question is really whether ICH2 or TR4, whatever dependent non-coding transcription regulation might be the part of active regulation for coding transcripts. And we wonder about that because a lot of genes that have this overlapping transcripts, coding, and antisense are regulatable genes. So we are looking into that, but we don't know that yet. So that comes down to whether there's biological functions of those transcripts. And given that, at least in TR4 single mutant, there are thousands of places that this happens. Some of them, I would be surprised if some of them are not used in that way. But I really don't think much has been done in terms of cryptic RNA analysis, partly because it's so tricky to do that.
So a couple of labs have shown that if you knock out acetylation
Mm-hmm.
in the coding region,
Right.
either by knocking out the small complex oxygen
Mm-hmm.
or by knocking out Z2,
Right.
this spurious transcription
Right.
in some genes,
Right.
real precise location.
Right.
Is that really at nuclear-free regions?
Right. Yeah, that's a great question. The question is, if you reduce, if you knock out some regulators such as RPD3 or SET2, you have intragenic non-coding transcripts. So we did wonder whether those start sites might be actually ORF NFRs. Unfortunately, those start sites have not been mapped very widely. So we actually speculated that in a paper that's under review, but that's a good possibility. We just don't know for sure yet. And also whether intragenic cryptic transcripts and intergenic cryptic transcripts are regulated in the same way. We actually don't think so because we don't have any evidence that intragenic cryptic transcripts are actually recognized by exosome. Why that is, we don't know, unless they go antisense. So really, a lot of interesting things need to be worked on cryptic transcripts. We don't even know why they actually recognize cryptic transcripts to start with. So those are things we're interested in. Yes?
There was a recent paper that by introducing some RNAi,
Mm-hmm.
I'm not sure about RDRP, if you can have RNAi.
Right. That's right.
I was wondering if those peptides somehow lose their RNAi that way.
Right.
There's a crosstalk.
Right, right. Yeah, it's a great question. So the question is, in Cerevisiae, there's no RNAi, but recently it was found just by introducing a couple of genes, you can actually recreate RNAi in Cerevisiae because they just recently evolutionarily lost RNA pathway. So, if the same thing happens in ISH mutant in higher eukaryotes, I'm pretty sure that RNAi goes all over the place. So that may be the reason why I-switch is not essential in yeast, but it is in any other organism. So the question is, if you introduced RNA system in Cerevisiae, what happens in I-switch mutant? And it's a really interesting thing to test. It's probably perfect rotation project, but we haven't done that yet. We are hands full right now, but I suspect that it will trigger massive RNA interference, and the cell will be in big trouble. But that's a really cool thing to try. And interestingly, POMB has a very strong RNA mechanism, and that's only the eukaryotes that don't have I-switch. So I wonder, having strong RNA mechanism may alleviate the need for I-switch and vice versa between Cerevisiae and POMB, but this is just speculation at this moment.